anyone that wants to see that, and then I can get started with the introduction. So I'm very much happy to introduce our speaker today. Again, she already told me once again how to pronounce her name, but, so I'm probably going to make it wrong, but Genevieve Konopka. She is originally from Pennsylvania, which was very important actually, this week, Pennsylvania. So she's currently a professor or associate professor of neuroscience at the University of Texas, Southwestern Medical Center in Dallas. So she did her PhD in neurobiology at Harvard, and before that she was receiving her bachelor degrees in brain and cognitive science in biology from the MIT. She did a postdoc with Dan Geschwind at UCLA, and then after a postdoc, she moved to Texas. So I'm very much looking forward to hear your story and what you have to talk about. I think you can go ahead and share your screen. And thank you again for joining us today.
Okay, can you see the screen?
Yes, we can.
Let's see if I can do the pointer. And the red pointer? Yep.
Yes.
Okay.
Yeah.
Okay, great. So thank you so much for the invitation again. This is exciting. This is actually my first virtual seminar, so we'll see how this goes. And I'm going to share with you our work on some of our cell type specific transcriptional networks related to autism. So I'll just give you an overview of my lab. We're interested in the genetic and molecular mechanisms of cognition, and in particular, what features of cognition might be unique to humans. And are these features related to the emergence of what we think of as human-specific disorders such as autism? So what is our approach to tackle this very challenging question? So we use comparative genomics, frequently comparing human to non-human primates. I'm not going to talk about that today. Recently, we've also been using measurements of human brain activity, either from fMRI or intracranial EEG. Also not going to talk about that today, but using these methods, we've been able to identify genes that we think are important for human cognition. Once we have the genes in hand, we can then do functional studies in cells. For example, human neurons. We've been recently working with iPSC-derived organoids. And we can also take these genes and do functional studies in model systems, such as the rodent. And then within both of these, we can carry out further transcriptomics and identify additional genes in this kind of feedback to really build these networks and hierarchical signaling of genes important in cognition. Today, I'm going to focus on some of our recent work on functional studies in rodents. And it's going to focus on some genes involved in autism. And as we all know, there's a strong genetic component to autism. I won't give specific numbers because that's a moving target. But two of the genes that we've really focused on in the lab that pertain to the genetics of autism are FOXP1 and FOXP2. And I want to just remind everyone some of the hallmarks of autism, abnormalities in social interactions and communication, as well as motor difficulties. And these are going to come into play as I describe our work related to FOXP1 and FOXP2. Okay, so what do we know about these genes in terms of their relationship to autism and other phenotypes? Well, both of these genes have been implicated in language disrupting disorders. Specifically, FOXP2, and I'll show you on one of the next slides, has been famous for its involvement in language impairment, and there's much increasing evidence showing its relationship to autism. It's also involved in this orofacial dyspraxia. FOXP1 mutations are also in humans shown to be involved in language impairment, autism, intellectual disability, which is not necessarily seen with FOXP2 mutations, psychiatric features, as well as other issues throughout the body. And I don't have it on the slide here, but there's some recent work actually from Simon Fisher implicating a third member of the FOXP family expressed in the brain, FOXP4. So that's pretty exciting for the field. Okay, so this is one of the most recent large studies in autism. whole exome sequencing showing if you so-called top genes associated with autism. And you can see here among the top 5 genes, FOXP1, and down here still reaching, pretty significant threshold, FOXP2. So in this large study, both of these genes have come up, and in many other smaller studies, we're seeing increasing evidence for both of these genes. So strong evidence for their relationship to autism. As I've already alluded to, FOXP2 is famous from this large family, the KE family, where there was an inherited verbal dyspraxia and shown to track with mutations in FOXP2. And there's been a lot of work by many groups in songbird, mouse, and in humans showing that while there are many conserved features for this FOXP2 family gene for example, frontal striatal circuits and vocalizations, we think that perhaps its role in the human brain might be involved in higher cognition and language, and hence this relationship to autism comes to the forefront. Now, both FOXP1 and FOXP2 are transcription factors. They can homodimerize or they can heterodimerize, and this heterodimerization will come into play towards the end of my talk. And what's really interesting about these genes, when you think about them with respect to their role in autism, is that if we take gene expression data from the brain span and look when they're expressed, hopefully what you can appreciate, their peak in expression here for FOXP1 in the striatum and here for FOXP2 in the cerebellum, they really have higher expression in this prenatal period. They're still expressed postnatally, but it seems like they're really expressed in the brain during development. And this is the time where we're seeing most of these autism genes really have enriched expression. So they're involved, we think, in setting up, the brain circuitry that becomes at risk in disorders like autism. And here I'm just going to summarize at a more detailed level where both FOXP1 and FOXP2 are expressed in a kind of mammalian or rodent brain. And so you can kind of see broad expression in cortex, hippocampus, striatum, thalamus, cerebellum, and hindbrain nuclei. But it changes depending on where you are. So for example, in the cortex, FOXP1 has expression all the way from layer 2-3 down here to the upper layer of 6, whereas FOXP2 has really this distinct expression pattern in layer 6 subplate with a few mingling cells here in lower layer 5. And this is important because this expression pattern also relates to the circuits these cells are involved in. For example, FOXP2 in corticothalamic projection neurons and FOXP1 in cortical or cortical striatal circuits. In contrast, when we look in the cerebellum, FOXP2 has really enriched expression in Purkinje cells. And I don't have it diagrammed here, but there is increasing evidence for FOXP1 expression in the cerebellum, definitely in deep cerebellar nuclei, also in Purkinje cells, but it changes and you have to look earlier in development and in the right place to see it. So it's not as prevalent as FOXP2. And one other brain region I just want to focus on is the striatum. We have 2 major cell types of spiny projection neurons there, the direct pathway or D SPNs, the ones that express DRD1, or the indirect pathway or the DRD dopamine receptor 2 expressing ISPNs. And there's much more expression of FOXP1 in these ISPNs and about more equivalent expression of both of these in the DSPNs. And this will come into play when I talk about our work in the from. And just to remind you, as transcription factors, these genes sit in the nucleus to regulate gene expression. There's been some preliminary work from many labs showing FOXP2 is regulating genes involved in, neuronal function, synaptic function, and also dopamine signaling. And I'll show you some of our further evidence for that. And FOXP1 is also involved in potentially things involved in neurogenesis. And there's evidence that early in development, FOXP1 is expressed in progenitors, also involved in synaptic function. But given everything we know about when and where they're expressed, there's still many things we don't really understand about their function in the brain. So how do FOXP2 and FOXP1 contribute to vocal and cognitive behaviors in a brain region in cell-specific manner? Because remember, I showed you that the pattern of expression is not all cell types that express them. What role do they play in brain development and morphology? What genes are regulated by them in the brain, and what genes are regulated by them in a cell type specific manner? And some of the work in my lab and from others in the field have shown that post-translational modifications of FOXP1 or FOXP2 can indeed affect the function. And so we're still just starting to dig into that. Okay, so just to outline the stories I'll present to you today, in part 1, I'll talk about FOXP2 in cortical development, and function in a cell type specific manner. And in the second half, I'll talk about FOXP1 in a cell type specific manner in striatal function. Okay, so starting with part one, FOXP2 in the cortex. So how might cortical FOXP2 mediate autism-relevant behaviors at the cellular and molecular level? And remember, FOXP2 in the cortex is primarily expressed in these lower layer 6 and just a bit in the layer 5 projection neurons. And all of this work was carried out by a very talented graduate student in the lab, Marissa Ko, who's now a postdoc in Oregon with Brian O'Rourke, so continuing to work on autism, and she's doing quite well there, I hear. So, and this was published late last year, so some of you may not have seen it, so I thought I would present it. I haven't had the opportunity to really present these data to a larger audience. Okay, so what did Marissa do to understand FOXP2 function in the cortex? Well, she used a conditional knockout mouse approach where she bred FOXP2 flox mice. These were mice that were generated by Simon Fisher's group and were available through JAX. And we crossed them to an EMX1 Cree mouse to really knock out FOXP2 early in development. So we had 2 lines. For controlled, we used flox animals and then the knockout with the Cree. And these are just some beautiful sagittal sections, immunostaining for FOXP2. You can see where FOXP2 is normally expressed in the brain. If you zoom into the cortex, again, you can see this expression in the deep layers, and in the knockout, you can see using the EMX1 Cree, while we still have FOXP2 expression in other parts of the brain, we've completely, again, in the zoom, knocked it out in the cortex. And that's what we were really aiming for with this knockout. Very low levels, undetectable perhaps expression in other areas where you might expect EMX1 to be working, such as the hippocampus. So one of the first things that we wanted to do was look at ultrasonic vocalizations. I'm sure many of you are familiar with these. These are calls that mice make either in the context of mating. This is typically the males make these, although females are known to make some calls. And also pups make these calls when they're taken away from the mom. They get cold and so they vocalize. And typically for a lot of autism models, this paradigm is used, but also people are using the adult vocalization paradigm. And because I've already said that in humans, variants in FOXP2 are associated with language and oral facial dyspraxia and all sorts of other things related to speech and language, we might have expected that we would have an effect on these ultrasonic vocalizations, particularly, some motor aspect perhaps of it. However, when Marissa measured adult calls in control and knockout, she did not find a significant difference. In addition, when she looked in the pups, again, in terms of call number, she did not see a significant difference. She did see a significant difference here in the sound registry of these calls. We know very little about the kind of mechanisms that underlie this in mice, and we're not affecting FOXP2 expression outside of the cortex. So we really don't understand what this means. And so we presented these data in the paper, but I leave that up to the field or someone later down the road in my lab to further explore that. But those are the data. And she repeated this. I don't have the N listed here, but it was quite a large number of animals. It was reproducible. So we didn't have an effect on vocalizations, but what about learning and cognition? Because again, we're focusing on the cortex here. So cognitive flexibility, we know, is impaired in children with language impairment, and developmental disorders like autism can involve learning impairments and behavioral rigidity. And so Marissa decided to carry out a water Y maze for reversal learning. So in this case, the mouse is placed in this Y-shaped maze and it's filled with water. And they learn on day 1 to swim to this platform for a reward. And this is repeated on day 2, repeated on day 3, and then later in the training, the location of the platform is switched. And then you have to measure how long it takes for the mouse to learn that now the platform is on the other side of the Y maze. And what Marissa saw is that both the control and the knockout could learn the correct location of the maze. And then here you can see it dip down for both control and knockout on the reversal, but the control animals learned significantly faster to switch to the other end. So this suggested that in the cortical FOXP2 conditional knockout that there was impaired behavioral flexibility. And so that was our behavioral phenotype that we had. And now, since FOXP2 is a transcription factor. Of course, as we're primarily a genomics lab, we wanted to understand what are the transcriptional targets of FOXP2 that might be important for this behavioral phenotype and perhaps other phenotypes that we just had not measured in this mouse model. And so we decided to use a drop-seq approach. And I won't go through this because I'm sure now, it's been 5 years since the mechar group published this paper. We're all familiar with it, but I just want to highlight a few key points about why we really like using a drop-seq approach. And we're going to use a commercialized platform, the 10x Genomics platform, for our experiments. One is that you get cell type specific expression, right? Because you're capturing in these droplets individual cells. You also can get absolute transcript quantification because you're including this UMI into your library preparation. And what's really nice is that while FOXP2 in this case is only expressed in a subset of cells in the cortex, we'll be profiling other cells. And so we'll see non-cell autonomous changes through this method. That's sometimes hard to see in a bulk approach. Those sorts of changes can potentially get washed out, or you can never be sure which cells are driving them. And so what Marissa did was carry out single cells. So these are cells, not nuclei. And we were able to use cells because we used early young brain, so postnatal day 7. These were actually, her conditional knockout mice were crossed to a DRD1 TD tomato because we wanted to isolate as much as possible the lower layer neurons. And so she dissociated the tissue and did flow cytometry for the TD tomato, and then followed by 10x. But hopefully what you can appreciate here from this UMAP plot is, for whatever reason, either through not having stringent enough flow cytometry controls or having expression of the tomato elsewhere, or low levels that might not be detectable at the protein level, but we can see that the transcript, or we can still pull it out. We saw expression, we saw most cell types from the cortex. So here we're seeing all of our excitatory projection neurons as well as inhibitory neurons. And it wasn't really limited to the lower layers. We saw some upper layer neurons as well. There was definitely, I think, a shift though towards the lower layers. Moreover, we could determine the percentage of cells in each cluster that were contributed either by the control or the knockout. And we had some significant differences in these contributions. So for example, in the layer 6 cortical thalamic projection neurons, where we know FOXP2 has high expression, we saw greater cells contributing from the control than the knockout. Those are the cells that we're primarily affecting. Moreover, we saw this also potentially compensatory increase in the layer 2-3 intratelencephalic neurons when we compared to the knockout cells. And a very cool result that Marissa got that was intriguing to us was in the knockout, we saw this increase in these migrating interneurons. This suggested to us a perhaps non-cell autonomous effect because we wouldn't expect FOXP2 to necessarily be expressed in these cells. And among the cells that are differentially expressed in this cluster were commonly studied genes with respect to these cells, so DLX12, SP8 and 9, and MEIS2 mice 2. We could also carry out a differential gene expression between the genotypes within a cluster, and we did see an effect on many genes involved in dopaminergic signaling. So the DRD1 receptor, PPP1R1B, which is downstream signaling of dopamine. And of course, we saw changes in FOXP2 expression, also some other interesting genes like FOS. But I want to say these were the cells we were targeting. And of course, you can see that some of the largest number of genes that are expressed are outside of this region, suggesting, again, this non-cell autonomous effect when you knock out FOXP2 early in development. So to summarize, FOXP2 directs autism-relevant cortical behaviors and gene expression in a non-cell autonomous manner. And I think this is a very interesting finding and something that I'm hoping my lab can follow up in future studies using some other time points to look at the role of FOXP2 in expression and also try and understand the implications, like how does this affect dopaminergic signaling? How does this affect the electrophysiological properties of these cells and potentially circuits? Because other work, for example, work from Pat Levitt's group and others have shown that using a similar approach to knock out FOXP2 in the cortex, there really don't seem to be effect on many of the projections out of the cortex. So why would we be seeing these changes in behavior that we observe in our mice? So there's a lot to be done. It could be at the cellular level in terms of physiology. Okay, so that was part one of the talk. And now where I'm going to spend most of my time is focusing on our work in the striatum, where we've been studying FOXP1, and I'll get a little bit to FOXP2 at the end, and I hope I can, be relatively within our time, so there's time for questions. Okay, so again, just as a reminder, FOXP1 is also a transcription factor, and within the striatum, it's primarily, it has expression in the DRD2 or the ISPN, and it shares expression with FOXP2 in the DRD1 or the D SPN. So we've got this kind of gradient of FOXP1 and FOXP2 expression across these common cell types, and this is just schematic to show the striosome and matrix components that really there's also that aspect of the striatum to take into account when one's thinking about the function of individual genes, and we did that indeed in this study. So apparently, from a number of papers that have now coming out, I'm just listing a few of them here, the clinicians are starting to call individuals with mutations in FOXP1 as having FOXP1 syndrome, because these individuals, as opposed to perhaps idiopathic autism where you really have a spectrum, they appear to have very similar features. And it could be at the end of the day, once we have a clearer picture of every individual with autism and know their genetic composition, that every single gene will become syndrome-like. But right now, there are just a few of these in the literature, and FOXP1 is among them. And so one common aspect across all of these individuals is that they, as you can see, have very distinctive craniofacial phenotypes. All of these individuals have language delay, and this was very intriguing to us because of this interaction of FOXP1 and FOXP2, and FOXP2 being this kind of classic gene associated with speech and language. All of these individuals have autism and or intellectual disability. So for the most part, as I've already mentioned, they're perhaps with the intellectual disability component, more severely affected perhaps than individuals who have mutations in FOXP2. And this quote I like to always mention when I'm thinking about these reviews of the cases is that it was noted that all individuals were delayed in achieving motor milestones. So why is this important? This is important when we think about where FOXP1 is expressed. And that is that it has this peak expression in prenatal development, in fetal development in the striatum. And we know that the striatum is a key part of the brain involved in regulating motor function. It's not the only part of the brain, but it certainly plays a key role. Okay, so a lot of the work in the field of autism and mouse models and studying has focused on the cortex, the hippocampus, a lot of interest in the cerebellum, of course, but there is definitely strong support for the genetics of autism to be intersecting with striatum and striatal function. So for example, this paper from Evan Eichler's group a few years ago showed that de novo mutations for genes associated with autism were enriched in genes that tend to be expressed in the striatum. In addition, some other work from Dennis Vitkup's group had similarly shown that autism genes or autism risk genes are genes that tend to be expressed either in these cortical layer 5, cortical striatal neurons, there's the striatum, as well as in both the DRD1 and DRD2 spiny projection neurons of the striatum. So here we see again this intersection of genetic variants and expression of genes in the striatum. So really supporting a role for the striatum. So a number of years ago when we first started working on FOXP1, really because initially I was interested in it because of its interaction with FOXP2, the first thing that we wanted to study was what we thought of as kind of a patient-relevant mouse model, which was a whole-body heterozygous mouse model. So if you think of the patients as being haploinsufficient for FOXP1, this might be the mouse you would go after. And this was work carried out by two very talented graduate students in the lab, Daniel Araujo, who's now working in the San Diego area as a scientific writer after he completed a postdoc, and Ashley Anderson, who's now a postdoc in Huda Zogby's lab. So proud of both of their progress through academia. And what they found was if they looked at the protein level of FOXP1 in the brain in either the heterozygous or the control, what was really interesting in the cortex for FOXP1 in this patient-relevant mouse model, we didn't see a decrease in FOXP1, even though it has high expression there. So suggesting it's really important in the cortex, and there's this compensatory increase of expression. And indeed, we have some other published work supporting that. But what Ashley and Daniel found was that there was at least a 50% or more greater reduction of FOXP1 protein in both the hippocampus and the striatum. The striatum seemed to be a bit more affected, if you will, just at the level of protein expression. And so, of course, because of this link to FOXP2 and speech and language, one of the first things we wanted to carry out was this USV, so the measurements of ultrasonic vocalizations in the pup isolation calls. And so what they found was that indeed, in contrast to what Marissa had found in the cortex, in this heterozygous mouse model, there was a significant reduction in the number of calls in with FOXP1 haploinsufficiency, or FOXP1 heterozygous animals. We also carried out gene expression. So this is bulk RNA-seq based on the date, quite some years ago now. And we then used this approach of weighted gene co-expression network analysis. We identified what we call modules of genes that seem to be significantly associated with genotype, so being a het for FOXP1 versus control. Two of these modules were very interesting because they had hub genes, or genes that were highly co-expressed, that seemed to be implicated, or known to be implicated, excuse me, in dopaminergic signaling. So again, here we have this DPP10, which is an accessory protein for a potassium channel, some calcium channels here, and then so ion channels here, and then in the lower module we found enrichment for things like DRD1A, so dopaminergic signaling here, and if you will, physiological properties. And so this suggested to us perhaps a mechanism of how loss of FOXP1 could be affecting cellular features that then play into behavioral phenotypes such as the ultrasonic vocalization. And so we wanted to really tackle this by doing some electrophysiology. I run a genomics lab. I don't do these sorts of experiments, but my good colleague down the hall, Jay Gibson, does. And so we teamed up with Jay to do these experiments. And what we had done is we crossed our heterozygous animals to reporter animals to mark the different two major cell types in the striatum. So these D1 and D2 spiny projection neurons. So marking the direct, or the D1 SPNs with the DRD1 TD tomato, and then just looking at intrinsic excitability, what we found was that there wasn't a significant difference in the hets versus control. If anything, in the hets, there was a tendency to be hypoexcitable in these experiments. In contrast, when we marked the ISPNs by crossing the animals to a GFP reporter and doing similar experiments, what we found was that there was a significant increase. So we found hyperexcitability when we have 50% or a heterozygous loss of FOXP1 in these ISPNs. And hopefully you recall what I told you is that FOXP1 has more, or if you will, relative to DSPNs, it's really enriched in the ISPNs. And we have very little FOXP2 expression in the ISPNs. So as I'll get to and probably say in one of the later slides, one of our working hypotheses, there's compensation here in the DSPNs by FOXP2. So the hypothesis is that FOXP1 regulates cell type specific pathways in the striatum, and that these might be some of the most vulnerable cells in individuals with FOXP1 syndrome. And so to tackle this and really delve into this further, Ashley continued on in the lab by carrying out a conditional knockout approach. And so we had conditional allele mice that we had obtained from Haley Tucker, similar to the constitutive knockout animals. And she crossed them to two different CREs, one using the DRD1 promoter or the DRD2 promoter to knock it out in this cell type specific manner. So in the end, she actually had 4 genotypes. So we had control flox animals. She had animals only using the DRD1 CRE to knock it out in the DSPNs, or to use the DRD2 CRE to knock it out in the ISPNs, or we also did the double conditional knockout. So knocking out FOXP1 in both D and ISPNs. And this is work that was published this spring, just as we all went into lockdown, summarizing what I think is really some beautiful work. So this is probably one of the first times I'm going through all of the latest details. So what we're interested as transcription factor in the genomics. So what are the cell type specific transcriptional targets of FOXP1? So Ashley did postnatal day 9 striatal single cell dissociation. Again, I'd like to emphasize that these are single cell data. So she made punches and slices, kept them alive like you would for electrophysiology, and then gently dissociated them. And we had 4 animals per genotype. And we used the 10x genomics system to do the profiling. And in total, after we did our QC and got rid of low quality cells, we were left with almost 63,000 cells across the 4 genotypes. And this is just a beautiful UMAP plot showing we were able to capture all of the major cell types in the striatum that you would expect at this time point. So you can see SPNs here, you can see progenitors, because again, we're at P9, you still have striatal progenitors. You can see non-neurons, oligodendrocyte precursor cells, astrocytes, microglia, and again, more SPNs up here and also interneurons. So there's actually a lot of data here, and we're putting together another paper where we kind of look at these data in the context of development, but for the purposes of answering our initial hypothesis, Ashley really wanted to focus just on the SPNs where we know FOXP1 is expressed. It's not expressed in interneurons. It's not expressed in astrocytes or other non-neurons. So we basically pulled out, subclustered only the neurons. In this case here, you can see the DSPNs, the interneurons. We included interneurons because sometimes they have somewhat similar expression patterns. And we also wanted to hopefully look a little bit at non-cell autonomous effects. We also have these ESPNs that had been identified in the mechar lab in the Saunders et al. Paper, and what Ashley's calling some immature SPNs. And here are just some charts showing the relative expression in each of these genotypes across all of the cell types. And hopefully what you can appreciate when we knock out FOXP1 in the ISPNs, what we found, or in the double actually conditional knockouts, what we find is this kind of increase, if you will. So you can see this increase in red in these 2 right plots. So that's an increase in the DSPNs and a decrease from the control and knocking it out in the direct pathway. So a decrease in the ISPN. So it seemed at the single cell transcriptional level that we were so-called losing the ISPNs and gaining the DSPNs. But because if any of you are doing these types of experiments, you know, the nature of profiling and sampling, we did some bioinformatic ways of addressing this by downsampling to show that these proportions didn't change if we downsampled and had exact same number of cells across all conditions. But we thought, you know, people still might not believe us, so maybe we needed to show this in another way. And so we did that. We went to the trouble and we crossed all of these genotypes to reporter mice that mark the D2, the ISPN, so the DRD2 expressing. And hopefully what you can see here, similar to these pie charts, similar to seeing this loss of blue in the pie chart at the genomic level, we also see this loss of green. So we're losing the DRD2 expressing, or the ISPNs. And here she quantified and showed that this is a significant reduction. Now, where are these going? Is it cell death? Is it cell fate? I mean, these are questions that we're currently exploring. I don't yet have an answer for that. We don't think it's cell death. And we did some other experiments to show that the timing of the 2 Cres are very similar within a half a day of one another. Those are very challenging time breeding across many alleles. So that's still an open question. But I think the take home from this is that knocking out FOXP1 in these 2 different spiny projection neurons at roughly the same time in striatal development gives you quite different phenotypes. And one of them is that there's this loss of the ISPNs. Okay, so so we had all this gene expression. What are the targets that we identified? Well, we found, even though we were losing ISPNs, we found many more differentially expressed genes within the remaining ISPNs. So not only did we seem to be perhaps losing some, but the ones that remained were quite different. And here I want to just emphasize that while we found a lot of autism genes that were regulated by FOXP1, and this was similar to what we had seen in our previous heterozygous patient-relevant mouse model, we also had seen a lot of known autism risk genes that were changing with loss of FOXP1. We similarly see this at the cell type level. And this is a very busy upset plot, but one thing that I wanted to emphasize other than the change in the autism gene is that we also see this compensatory change and increase in FOXP2. So this goes back to this idea that I said that FOXP1 and FOXP2 might be having kind of this interplay or this compensatory expression for one another, because when we had done the electrophysiology experiments, we only saw changes in the ISPNs, not the DSPNs. Again, we see this at the gene expression level. The DSPNs are relatively intact. And could this be due to their normal relatively high expression of FOXP2? And even though the ISPNs are trying to compensate by upregulating FOXP2 here, it's not sufficient, but it may be helping the ones that are remaining to a certain extent. Okay, so we had made these conditional knockout mice, and so we wanted to see if they would have any behavioral changes so that down the road we could start to link the gene expression changes to physiology, circuits, and behavior. So Ashley put the animals through a number of behaviors that one might do for a striatal conditional knockout mouse. So one would be the rotor rod, the accelerating rotor rod, where the animal learns over several tries a day, over multiple days, that it needs to stay on the rotating rotor rod. And then you can calculate how long it takes for the animal to fall off the rotor rod. Hopefully what you can appreciate, the control, and again, these DSPNs or the DRD1 expressing conditional knockout mice are very similar, but we did have a significant reduction, or we had a significant effect on rotor rod learning in the DRD2 conditional knockouts as well as the double conditional knockouts. So you can see this here as well. We also wanted to see if this was due to some sort of, just were there general motor deficits. She did other things like grip, those seem to be fine. But also in the open field, we didn't see a deficit. In fact, we saw that the DRD2 and the double conditional knockout mice had increased velocity. So if anything, that wasn't likely affecting their performance on the rotor rod. We also, in addition to motor behaviors, we wanted to look at social behaviors. So again, we carried out these PUP USV analyses. And now in contrast to the rotor rod deficit, where we saw the ISPN conditional knockout mouse having a significant effect, in this case, now we're seeing the DRD1 or the DSPN conditional knockout mice having a significant reduction in number of calls over development. We also carried out measurements of nesting, which we think is a socially relevant behavior. And here so this is just to show you what it would look like over time. Here's a snapshot of what the nests would look like in our mice. And this is quantification across many animals. And we found a significant reduction in nest building, again, in the D1 or the DSPN conditional knockouts, similar to how we saw an effect on the USVs. But in this case, in contrast to the USVs, we actually saw a significant reduction in the double conditional knockout mice. So this is really interesting to us, this contrast of behavioral deficits when you knock out FOXP1 in the D1 versus the D2 SPNs. We wanted to determine if loss of FOXP1 was affecting projections. Remember, I had mentioned work from Pat Levitt in the FOXP2 cortical knockout mice that didn't really seem to be projection deficits. And so we took all of our mice, we crossed them to double reporter animals, and we did this analysis using whole brain imaging and could quantify projections. And here's just a little cartoon summarizing these. And what I want to draw your attention to is that in the D1 SPN, so the D1 conditional knockouts, we didn't have an effect. Here's where we're knocking out FOXP1 there. We didn't really have an effect on any of the projections. In the DRD2 or the ISPN conditional knockouts, not only do we see an effect on projections, out of projections here where we're knocking out FOXP1 into the GPE, we're also seeing an effect on projections in a non-cell autonomous manner in the other cell type in their projections to the GPI. So we're affecting both. So we had the gene expression. So we asked, what could be some of the genes that might be having this non-cell autonomous effect, and are they interesting? And indeed, some of these genes are known to be involved in, say, projections or, linking of cells to one another, such as cadherins, contactins. So there are some potential targets that we could follow up in the future to really understand some of these projection defects. And then we wanted to see in the conditional knockout animals if the results would be similar to what we had seen in the heterozygous animals. And indeed, if, again, collaborating with Jay and a postdoc that we both co-mentor, Nitin, we carried out similar measurements of intrinsic excitability. And if anything, it's an even greater effect, this increase in excitability in these remaining ISPNs in the conditional knockout. There's also changes in resting membrane potential and input resistance. You can see this change. And what's really nice is now we can use the single cell data to identify potential candidate genes for what might be regulating these changes in intrinsic excitability, and in particular, Nitin and Jay have been focused on a number of potassium channels, and they actually, I don't have it here, have a manuscript in bioRxiv that's under revision, where they've documented and done really beautiful and detailed electrophysiology, trying to separate the contribution of various channels that are affected in these conditional knockout mice as well as in the heterozygous mice. So I think it's a really technical tour de force, so I encourage you to check it out. Okay, so back to this in my last 5 minutes or so, this idea of, why are the ISPNs so prominently affected? Why are the DSPNs seem to be resistant? And this idea of perhaps there's compensation at the level of FOXP1 and FOXP2, they can kind of regulate expression of one another, or one come in some manner that we don't quite understand. But also we know that they can heterodimerize to regulate gene expression. So it kind of makes sense that they may be able to monitor the levels of one another if this heterodimerization is happening in vivo and also homodimerization. So that's an open question. We don't know in a cell type specific manner, when are these two transcription factors heterodimerizing, what are the targets versus homodimers of each one of those. And the cells we'd want to focus on are the DSPNs, or the DRD1 expressing, because that's where both of these tend to be normally expressed. I think this is a super challenging project. It's something I wanted to do for many years, and why I got interested in getting the FOXP1 mice and working on them. And somehow I managed to convince a very brave graduate student in my lab, Nawaz, to undertake this project. Okay, so how is Nawaz tackling this? Again, just as a reminder, the DRD2 expressing cells primarily express FOXP1. It's the D1 expressing spiny projection neurons that have both FOXP1 and FOXP2. And so he took a DRD1 CRE, and he's making double conditional knockouts of FOXP1 and FOXP2 in DRD1 expressing neurons. So in these animals, you'd have 5 alleles. So you'd have the double FOXP1, double FOXP2, and the CRE. Okay, so again, focusing on genomics, Nawaz has carried out a series of, in this case, we've kind of switched to nuclei just because of the way the genotyping works. We can never, we wanted to minimize some of the batch effects for the library prep and things like that. So we switched to, even though it says cell, it's actually nuclei, single nuclei RNA-seq from this DRD1 conditional knockout mice. And so he's generated a lot of data. These are just some of the data from adult animals. We also have from the same age that I showed you from Ashley's data set from around P9. And this is not showing you all the data. This is just a subclustering of the SPNs only, where hopefully you can appreciate, we get the DSPNs, we get ISPNs, and here we're seeing those a little bit unusual elusive ESPNs, which I don't have time to talk about today. And this is just a table showing you where we are in terms of UMIs and genes and things like that. And we're trying to add another FOXP2 only conditional knockout to make these even N of 3 across all. But I would say that we've been analyzing these data for the last 6 months during quarantine. And I would say that, the preliminary data look really interesting in that, as one might expect, in the double conditional knockout mice here, we see more genes differentially expressed than we do with either of the single knockouts. We were surprised that there weren't more genes changing, but these are adult animals, so there's a lot more time, I guess, for compensation to occur. We know from the preliminary data in the P9 animals, there are many more genes that are differentially expressed. But in terms of trying to understand within the context of autism, many of the targets that are affected or that are changing in the double conditional knockout mice are genes that have already been implicated in autism. So again, showing that these transcription factors that themselves have variants associated with autism also tend to regulate gene expression programs that are affected in autism. And then we can do enrichment, and indeed, they're significantly enriched for both autism genes as well as FMRP and synaptic genes. So that's really great. And then NWAS has also carried out some really beautiful behavioral data. It's challenging because to get all of these alleles in place. But I think that we're now getting up to a reasonable number of animals. And in the rotarod, what he's found is that compared to control animals, neither of the single conditional knockout mice have changes relative to control. It's only when you knock out both of them in the D1 expressing cells that you see the significant effect on learning in the rotarod task. And I just like to remind you, in our previous study, this is in agreement, because the D1 animals did not have an effect on the rotarod. It was the other ones, it was the ISPNs. The D1 knockout animals seemed to be okay, and it was this hypothesis about FOXP2 compensation. Indeed, that's now what we're seeing borne out in this behavior. Similarly, in the open field, looking at just here plotting velocity, neither of the single conditional animals have changes relative. We have two different controls here. Not only do we have the double flox animals here, we have Cree-only controls. It's only, and again, we had seen effects, remember, on the ISPN, not the DSPN. We thought there was compensation. And indeed, here we see when you knock out both transcription factors, now you unmask a phenotype, again, suggesting that there probably is compensation by both of these. And then in nest building, we were able to repeat Ashley's finding that in the D1 animals, there's an effect on nest building. And now when we knock out FOXP2, there seems to be even more synergistic effect on this behavior. So again, I think taken together, the gene expression and the behavioral data really support this idea of FOXP1 and FOXP2 working together and compensating for one another in these DRD1 expressing spiny projection neurons. So here I'll just like to summarize the results of the STRIME, since I spent most of the time on that. The FOXP1 cell-specific genetics have identified loss of ISPNs here in blue, and also ASD-relevant behaviors. And we know that it's important for these ISPNs to regulate motor coordination and hyperactivity. In contrast, in the DSPNs, it appears that FOXP1 is important for regulating social vocalizations and nesting behavior. And now with these new data that NWAS has been generating, if we make these double conditional knockouts that includes FOXP2, we're unmasking the cooperative role of FOXP1 and FOXP2 in these DSPNs to regulate both motor coordination and hyperactivity. And so just to summarize the work I showed you here, and other work from the lab, we've really focused a lot in my lab on these two transcription factors. Today I showed you cortical Foxp2 mediates cognitive flexibility and social behavior. It affects interneuron development and upper layer gene expression in a non-cell autonomous manner. And that FOXP1 mediates cell-specific gene expression relevant to autism and also gene behaviors in a cell-specific manner. And we have this compensation of FOXP1 and FOXP2. We also have published work on simulation of FOXP1, mediating cortical migration and dendritic outgrowth, and that cortical FOXP1, in contrast to cortical FOXP2, is critical for vocalization and social behavior. And we've also been very interested in the cerebellum, and hopefully in the future we'll continue to do that. We've shown that simulated FOXP2 mediates vocalization and motor function and regulates dendritic outgrowth and gene expression. A lot more to be done there. And so I just would like to thank Jay Gibson, my ongoing collaborator with all things FOXP1 and now FOXP2 electrophysiology. He's been a great collaborator. I think I highlighted throughout the people in the lab who've worked on this project. Here's just a fun Zoom lab meeting from the summer. And of course, funding, a lot of important funding from the Simons Foundation and NIH to support this work. And I tried to end early, so I'm happy to take any questions about this work. And thanks for the people who are watching.
Thank you very much, Genevieve. That was wonderful. I don't know if you want to unshare the screen so that people can see you. I mean, questions from the audience, you can actually raise your hand, but you should also be able to unmute yourself, but maybe I call you first. So please feel free to ask questions. Maybe I start. I was wondering, so on the first part, but this is probably also applies to the second part. So you're using conditional knockouts, and for the FOXP2 deletion in the cortex, you have seen also non-cell autonomous effects. And so I was wondering how many of those are contributing to the phenotype, and whether, in the case you would delete the gene also in those other cells, so let's say interneurons, whether the phenotypes would be the same, or in fact, some of the phenotypes that maybe emerge from those cellular, I talk about behavior phenotypes mostly, of those non-cell autonomous effects are an effect of the fact that you're deleting them just in a cell type.
Yeah, I mean, so complicated. So, you know, the thing that's been intriguing to us is that the non-cell autonomous effects in gene expression we see are in cell types that don't normally express these transcription factor, right? So is it through some sort of signaling, or is it that because, for example, FOXP1 is expressed really early in development in some sort of progenitor, and, you know, our EMX1 CRE really targets it early, could we be affecting the progenitor pool in some manner that's leading to these changes? So I think, you know, to figure out the contribution to behavioral effects, you know, how could we do that? I mean, I guess we could try and replace expression of one or the other in certain cell types, or misexpress expression in those cell types in some way, and at least the genes that are affected, the targets, and see if that rescues the behavior. I've never been really, other people, I think probably you and other people, you know, they have these beautiful rescue experiments where they can figure out a target and put it back in, and then the behavior goes away. We've never really been lucky in that regard. Maybe we're not guessing what's the most appropriate target, or we're not thinking about it, you know, deeply enough. But I think it is interesting. Like, is it the change in the migrating interneurons that's leading to the abnormal cognitive flexibility in the FOXP2 conditional knockout, or is it the change in the lower layer cortical thalamic projection neurons? Because we know both are affected. And I think we'd have to think of, maybe we could do something with optogenetics too. I don't know. I mean, that's a little bit outside my realm. All I can think about is kind of gene replacement to try and tease it apart. Or we also, what I'm trying, you know, to get funded is, you know, doing manipulations, inducible conditional knockouts at different time points to really see when things are affected, what cells are affected, and then how does that read out at the gene expression level? How does that read out at the behavioral level? So much to do.
Yes, as usual. I mean, that's good, actually. But because you mentioned optogenetics, this brings also to the other question I had. Mostly thinking about the second part of your talk, but I think this is also applicable to the rest. So I was wondering whether the fact that now you are kind of associating specific behaviors with different cell types, or receptor expressing cell types, so different cell types, whether these you expect that reveals in general the function of those cell types and the circuitry, so that kind of you use those knockouts not really to study the disorder itself, but in fact to really study the cells responsible for those behaviors, or how much of those you expect that instead are more like could be a gain of function that is not naturally so expressed by those circuitries. So what do you think about that?
Yeah, I mean, I would say in general, I think that our ISPN behaviors that we saw with loss of FOXP1 would be typical of like any sort of bad thing happening to ISPN, whether that was through, you know, optogenetic shutdown or some sort of drug. I think it's really within the the context of the literature of how we were affecting the indirect pathway. So I think it was pretty expected. We have actually generated a lot of data that never went into the paper, other things like adding cocaine and looking at other behaviors. And it was really, you know, on par. I think the only thing that was unexpected was when we were doing the circuit projection neuron mapping and we saw that non-cell autonomous effect, because usually when people are manipulating D versus ISPNs, you know, they're not normally going to get these non-cell autonomous effects. So this really suggests to me again that, you know, FOXP1 and potentially FOXP2 are regulating genes that affect things that get signaled or secreted somehow to the, or, you know, it could be really complicated where it goes through the normal indirect pathway and then it's a feedback all the way back down through and affecting the indirect pathway, because we have enough time in development in theory for that to be the case, because we only did the imaging on the pathways in adults because of the registry. We had to register to the Allen Atlas, if you will, and they only had an adult one for this sort of analysis. So we don't know what's going on when in development these projections are being affected. But in terms of the behavior, I think it was pretty expected. That what wasn't expected, I guess I would just like to end on, was, you know, the USVs. Like we didn't know what to expect when we knocked out D versus I. And so the fact that we only saw it in the DSPNs was a bit surprising. But then when we started building this theory with FOXP2, we're like, oh, maybe this makes sense. I don't know. Still more to be done there in terms of the vocalizations, I think.
Maybe, I mean, the last question from me. I mean, this compensatory effect is very, very interesting, right? And I guess that you are looking more into the mechanism, really, how this is regulated. So how basically one can sense that the other is missing, and how you can upregulate the levels of the other eventually. Do you know anything about that?
I wish I did. I do know that FOXP1 self-regulates. I don't know, we've seen that, and other people have shown that it kind of regulates expression of itself. It's possible there's literature on that that I've somehow missed, but I don't think there's any direct evidence for one regulating, you know, like directly binding and regulating the other. And that's, some of that stems from the fact that the antibodies for these transcription factors don't really work well for chromatin IP studies. So we can do things like ATAC-seq and things like that to look at chromatin accessibility when we change them, but that's not really going to directly answer the question. We're doing I guess what I didn't say is we're doing cut and run. Nawaz is trying to do cut and run in the brain tissue for both of these. So in that case, we're hoping to get around the antibody issues and address this more directly. But he's still working out optimizing conditions, because I'm not aware of a paper that's been published using it in brain tissue, but I could be wrong. I think it's just, everyone wants to do it and it's really hard.
Great. Then I think I thank you very much. Some of the people were sharp at the time and start leaving, but so thank you very much again. This was a really beautiful work, and it is a beautiful work. And thank you so much for participating. And I hope to see you soon in New York again, as we were used to do.
Yeah, definitely. Thanks so much, and thanks everyone who showed up. Happy to take questions too by email if anyone was shy and wants to ask later.
So as I mentioned, this was streamed in YouTube, so I will share also the channel with you and anyone else that probably will watch it later.
All right, awesome. Thank you. Take care. Stay safe, everyone. Okay, bye.